In your final exams or terminal tests and the like, you may be asked in practical biology, given a sample, given materials to test the presence or the absence of a particular food substance. Or else you can be asked in agriculture theory, that's an essay, like to describe an experiment of how you can find the presence or absence of a particular food substance. So this video tikambira napo some of the tests which are conducted to determine the presence or absence of particular food substances. So let's now be together to tithandizana motani. Now, when we talk about a food test is like laboratory procedures conducted on foodstuffs in order to find out the presence or absence of particular food substance. That's the food test. And the chemicals which we use to determine the presence or absence of that particular food substance, we call them reagents. So reagents are chemicals which we use. So in those reagents we include iodine solution. We use iodine solution. We may also use dichloropheno, which is DCPIP, dichlorophenoindophenol. We can also use Sudan 4 or 3. Yeah, those are some of the reagents which we use to determine the presence of particular food substance. So let's now start our business. We should start on by testing for reducing sugars. Testing of reducing sugars. That's our first thing. How can we test for reducing sugars? By the way, we should now start discussing what are the reducing sugars. When we talk about reducing sugars, these are sugars which remove oxygen. Which remove oxygen. They remove oxygen from Benedict solution. That is why testing for reducing sugars. And I'm going to Benedict test. That's Benedict test. And examples in the glucose. Mostly we talk much on glucose when we talk about reducing sugars. That's glucose. So testing for reducing sugars and testing for glucose. My guy. So procedures sinimalemba notes izo muzongomvetsera bwino. So procedures imachitika, we have to prepare food solution. Ngati food sample yathu iliyouma, we have to dissolve it with water. For instance, in the ufa ndikuti tikwenera kufewetsa ndi distilled water kandi chinangwa, we need to crush it and add distilled water in order to obtain the food solution. Whatever the way, but we have to obtain the food solution. And then the next thing we need to do, we add 2 cubic centimeters of Benedict solution. And then chimene tikandi test chibwatu many mumutika mu solution ife containing the food, and then tizapanga add this guy kwa panga add minimum, and then we heat gently. Timapanga heat in water bath. So timakhala ndi kaijeni kenet in water bath like this, ndikuti mulimadzi minimum. So kupanga applying heat like we don't heat directly to avoid explosion. You know, these chemicals sometimes they can explode, so we heat them in water bath. They timapanga heat pang'ono gently pang'ono, and then we observe the color change of the solution. Mix ya twitch. Kuti lidzo kuti minimum reducing sugar kali mafunika ikhale lady precipitate. Timapanga form lady precipitate precipitate kapena brick red. If the solution changes the color from blue, normally Benedict amakhala blue from blue to red precipitate kapena from blue to brick red, ngoza kuti mmene momwe muli chani muli juicing sugars. But if it doesn't change any color kapena kuti yangokhalabe blue like that, what it means is the food substance does not contain reducing sugars. So opposite of reducing sugars, that's non-reducing sugars. So let's now see how we can test for non-reducing sugars. Testing for non-reducing sugars. Chambilira ajamanina kuti they remove oxygen from Benedict solution. From these guys, they don't remove oxygen. Now examples of those sugars and disaccharides. So disaccharides include this one, the most famous one, sucrose. That's a sugar from sugar cane. Sugar mina vezeka muzimbe. So that's the thing. And now procedures amene matsata chimene chimachitika, we have to obtain the food solution. Let's say kuti atipatsa nzimbe kuti tipange test for non-reducing sugars. We need to crush them. We need to crush it using a mortar and a pestle. We kupanga squeeze to obtain the food solution. And then chimene timapanga tikazisiya zinthuzo in our test tube. So that's our food solution. That's our food. And then the next step we need to do, we add some drops of diluted hydrochloric acid. Diluted, ndikuti tikapanga ku add ukuku tizapanga add HCl, hydrochloric acid. So so tipanga add HCl there. And then we're going to boil this. This is boil. We are going to apply heat here. Chilenga choti this hydrochloric acid plus heat amakapanga break the bonds of sucrose. Pajamana kuti it's disaccharide. So disaccharide, it's a combination of monosaccharides, like monosaccharide plus monosaccharide plus monosaccharide plus monosaccharide. So this is disaccharide, that's the disaccharide. So heating will break the bond that exists between the monosaccharides. So the purpose of heating the food sample in presence of hydrochloric acid, that's to hydrolyze the hydrosaccharides into monosaccharides. So that's the thing. When they to break the bonds. And the next step, we cool. We ready the mixture to cool. They would move this into zigazirani. Zizire. Zigazira. Chimeti mapanga tsopano. You're going to add sodium hydroxide. Sodium hydroxide, this one it's a base. If you add this one, akapanga reacting the HCl to neutralize the acid. So the purpose of adding sodium hydroxide, that's to neutralize. And then the next step, timangopanga follow procedures for reducing sugar. We are going to add 2 cubic centimeters of Benedict solution. We're going to add 2 cubic centimeters of Benedict solution. Mubika mono muja. So this one, that's our beaker, and here is my food substance, and the hydrochloric acid has been neutralized. And then I'm going to add the Benedict solution. And after adding Benedict solution, I'm going to heat this banga heat. in water bath ndipanga heat in water bath bwino ngati mene mapangira when testing for glucose. This panga heat kwina kundumatani ndimapanga shake at least for 5 minutes. Basi heat basi kwina ku shaking gently and then observe the color change. If the color changes from blue to brick red or red twisted, the food sample contains non-reducing sugars. So that's the stuff there. That's how we can test for non-reducing sugars. Those are the steps. And if you're enjoying this video, please don't forget to subscribe. I'll be telling you to subscribe in each every video I make. So, guys, subscribe. Thanks for that. And if you didn't, please subscribe. You can share the videos. You can also make comments on the videos. I need your suggestions as well. Also, you want to contribute, you can send me anything so that I should upload more videos for you. And the next thing we're going to test for starch is a test for starch. In testing for starch, this one is one of the famous tests. So the reagent which we use iodine solution. That's the one we use. So procedures as usual. If our food sample is solid in form, we are going to crush the food sample, add distilled water to obtain the food solution. Mind you, it's not always that you'll be crushing the food. It depends the food sample which have been given. No need to say crush the food sample. No. So avoid such things. And I remember last time okay, forget about that. It's physics. It's a physics example. So I'll give you that example. And physics, that's motion. So that's the first procedure, obtaining the food sample. Obtaining the food solution. And the next step, we add the few drops. We add a few drops of iodine solution. That's our reagent. Iodine solution. So consider this as our test tube, and we have our food solution here, and then we are going to add few drops of iodine solution. So some few drops, 2 or 3 drops, and then we observe the color change. We just observe the color change. If the color changes from brown, from brown to blue black, color change from brown. I think I should use another thing. From brown to blue black. That's blue black. And some other books remember dark blue. They go to starch alipo. They go to the starch is present in that food sample. But if the color does not change, iodine solution is brown in color, and if it doesn't change and it stays brown like that, the food sample does not contain the starch. So that is the thing. And sometimes the color can change from orange to brown, like magungo sinta bangono kumano kali mafunika isintelete yonse kale blue black. So that's the thing there about testing for starch. I remember in 2015, yeah, 2015, anatibweretserabo sugar cane. Komanso anatibweretserabo cassava. Komanso there was this hiding solution. Atoza kuti tidule nzimbe uja cross section, komanso tidule chinangwa cross section, and we should add a few drops pa chinangwa, we should also add a few drops pa nzimbe. Noona kuti chinangwa chija chinasintha solution ija inasintha kukhala blue black, pomwe nzimbe kuja kale nangokhala chonchija ya iodine. Tanthauza kuti chinangwa chili ndi chani, chinangwa chili ndi starch. So that's the thing. And the next thing we need to discuss here is testing for proteins. In testing for proteins, what we do there, we use bullet solution. Bullet, spelling, bullet. So that's bullet solution. That is why sometimes testing for proteins it is known as bullet test. Bullet test. It's the same thing. So that's our reagent and the procedures there. Our procedures. The first thing, to obtain the food sample. Not food sample size, but food solution. Take the food solution. If your food sample is in solid form, you have to crush it, add distilled water to obtain the food solution. And the next thing we need to do, we're going to add some few drops of bullet solution. Some few drops of bullet solution, and then we shake. Mavanga shake you shake gently. Mavanga shake gently and observe the color change. Mavanga observe the color. So let's say this is my test tube, and this is my food sample. That's the solution. The solution containing the food, and I've added some few drops of bullet. Some few drops of bullet there, and then you are going to shake this. I'm going to shake gently, and then observe the color change. Now, what we're supposed to change? The color is supposed to change. Color should change from blue to that's violet. Kapena purple, violet or purple. Bullet solution, I'm a color blue in color, but in presence of proteins, the color changes to violet or purple. So that's the thing. But if the color does not change, it means it stays blue like that. It means the food sample does not contain proteins. So that's the thing. The other method, some books. I remember we're supposed to have our food solution. We're supposed to have our food solution there, and then we add 1 cubic centimeters of sodium hydroxide. 1 cubic centimeters of sodium hydroxide to the food solution. And then mpanga shake bwino. After shaking, and then mazapanga add 1% of copper sulfate solution, and you should observe the color change as well. So makala changes zake makala meal. So to makala ma steps angapo. Combination of this is the same as bullet. So that's the thing. And then the next video is about testing for lipids. Not in the next video, but it should be the next slide or whatever. But I'm gonna focus much on testing for lipids. The other test is test for vitamin C. And in testing for vitamin C, tumokitsa ntchito reagent yochedwa imadi use the other color. It's a dichlorophena, endorphina. So in short, it means D, C, DIP. That's in short dichlorofene and endorphin. So for this one, I'm a color blue in color in general. But when there is acid, I'm a simple color red. But acid. However, some other chemicals like let's say vitamin C, kali masimha kutoka ku blue kuza colorless ama colorless. So let's say we have this guy lemon juice mukhandi test kudano ndi mukhali ndi lemon juice ndi mugwetsere mu DCBIP wanu amachoka ku blue and kusintha kukhala colorless whenever there is vitamin C. So lemon and those are some of the fruits which contain vitamin C. So that's the thing. So njira yake ndio mweja ngati mimi tunaongera zinazi crushing the food if it's solid to obtain the food solution. Kena kunoa panga mix the mixture with the dichlorofene and endorphin, which is DCBIP. General color is blue, amasindo colorless. Accountable for acid, amasindo color red. So that's the thing there. My guide. In testing for lipids, we have 3 ways. We call that one as spot test. What we need to do, we just drop our food sample on the plain white paper. The paper should be white. So you just drop that the food solution. Got the food really solid form. We just lab it on the white paper. After labbing or after dropping the food solution on the white paper, you just let it to dry, and then you direct the paper to the sunlight and you observe what you see there. Possibly, what you see is called translucent. Translucent, like some spots like that. A grounded nuts. Mukupanga lab with the sample. After labbing and direct the paper to the sunlight, this is the same result as well. So it depends what the food sample is it in solid or it's in liquid form. So that's the thing, spot test. And the other way, we call that one as immersion test. In immersion test, this one will apply law of solubility. We know that lipids are insoluble in water. Lipids are insoluble in water. We know that we can form 2 layers in water. But the lipids, they dissolve in ethane. So lipids are ethane. Could we test if the substance contains lipids? This is our beaker and this is our food solution. We're going to mix it with the ethane. Mix in the ethane and dissolve. You're going to add water. To confirm what is into is dissolve. And dissolve, water should turn into cloud. But there is something on top, like a cloud. Like something like that. Cloud in nature. There will be minimum facts. Our food sample, our food solution, lipids. Komangati pango khala clear opanda china chilichonse ndiye kuti food sample yaife ilibe lipids. So that's the thing, immersion test. Njira ina number C, timagwiritsa ntchito Sudan test. Sudan test. we apply that one, Sudan test. So what we need to do, what happens in Sudan test, get the mukandi test cube, and in your test cube mukandi food solution, food test, from Ivangi, and then we add Sudan 4. That's a Sudan 4. I don't know, could you not do Sudan 4 again? Move and add some few drops of Sudan in there. Then you can Sudan is like a diet. It's a dye which stains in lipids. Amatsalira in lipids, amatsalira. Makawuri, there is no lipids. I mean, the food substance does not contain lipids. The dye mavanga accumulate on the bottom. So let's say that this is the bigger dye accumulates at the bottom like this. Zongobanga accumulate kupanga sink. It is sink under the test tube, and this is our lipid. The solution I food solution ilibya lipids. Kumanika kuri zindu zijya zatsalira izabanga stain like this. Let's say this test tube data gets mu study test life then solution life is zibanga stain in lipids. Never minimum lipids. So positive is that it's stained. Lipids is stained. Akaburi izindu ziriya muri lipids ziriya zibanga accumulate at the bottom, they sink. So the dye sinks at the bottom. It sinks at the bottom. That's about Sudan testing for lipids. And this marks the end of my video, and that's what I prepared for you. If you have enjoyed the video, don't forget to subscribe. Also, if you want to contribute something so that I can upload more videos for you, you can do the same. That's the number 882-435-562, and your support will be greatly appreciated. So that's it for this video. Thanks for watching, and have a lovely day.